Good afternoon, everybody. I'm really excited to be here today and tell you a bit about what we've been doing at Deep Mirror. And Deep Mirror is like the intention of the company is like a lot of things that everybody's been speaking about today, like all these novel technologies for in life sciences and AI and bio and chemistry. We're trying to take all these technologies and bring these technologies to an end user. So like for drug property prediction, this is medicinal chemists, right? And medicinal chemists probably can't train some of the models that have been discussed today. So how can they start utilizing these technologies? And I'm going to talk today about some of the findings that we've made when trying to serve these models up to users in the lab. And specifically, we focus on drug discovery. And drug discovery is, or like bringing a drug to market is a super expensive process. So it can take 10 years, can cost billions of dollars. So like when making decisions in this, it's good to have good models to make predictions. So what's happening at the moment with AI and drug discovery? So here's some articles that I recently found. So one's from May, one's from the other week. So new superbug killing antibiotic discovered using AI. What was the technology behind this? Well, this was graph models that Pietro's just spoken about now. And then we have another one here. A biotech begins human trials to drug design by AI. So this is all the research that's been discussed now is being translated in industry by big companies. But how do we make this more accessible for smaller companies? And like why? Why are people starting to do this? So when you're developing a drug, you start off with a molecule, and you may want to predict certain properties of this molecule. So this could be activity, so this could be the way that it interacts with a protein, or you might be interested in biological molecules. And during the development cycle, there's anywhere between 300 and 5,000 molecules made and tested in these cycles. And this is a lot of time and resources that go into making these molecules. So one place where machine learning can really help is predicting the properties so you can screen molecules without having to run these expensive lab experiments. And they're expensive for two reasons. One, you have to synthesize, and you have to go and do a test. So if we can minimize doing that, hopefully we can accelerate the cycle of drug discovery. And like just to comment about like why do we want time and efficiency savings? Well, like at the end of the day, this is drugs that are going to patients. So if we can de-risk faster and screen things faster and understand the properties and move faster, we can get drugs to patients. So like how do people do this at the moment? So as a company, we've been going for a couple of years now, but we found it very, very difficult to figure out like what machine learning technique does actually enable you to build the best model. I mean, in Pietro's previous talk, he must have mentioned 5 or 6 different graph architectures. So which one should you build on client data to predict a property? But that's just graphs. There's also expert features. So this is like extended connectivity fingerprints that some people may be familiar with, which is more of a heuristic and a deterministic algorithm. And so you could choose expert features, you could choose graph features, or you could choose language features. So taking the SMILE map representation of a molecule, using some form of large language model to create some embedding space, and then building a model on top of this. So like with everything in the literature, some papers say, oh, graphs are best, use graphs. Other papers say, language models, use them. And then there's other papers that say, don't do deep learning. These classical methods are better. So we wanted to get to the bottom of this as a company. So when we interact with clients, we can serve them up the best model for the task in hand. So now I'm going to talk a little bit about how we went about this. So first, I'm going to start talking about data sets. And there's quite a few public data sets that are classification problems. So this would be like, it's the question of, is this drug active, yes or no? This, from our experience, is really not useful in real-world applications. And The reason is most times where we've seen machine learning utilized, it's from predicting a value and sorting a list and prioritizing a list and moving to the next step. If you have active or not active, it's very difficult to get a sense of prioritization. So we purely focused on regression tasks for this. And we took some really common public benchmarks and some internal data from some of our clients. So TDC is one of them, which is a fairly new curated one. This mainly focused on ADMET properties. So this is like the biological properties of the molecule. MolNet is super famous for benchmarking a lot of the graph architectures. And then there's another one called ACE. It's our internal abbreviation, but this is activity cliff. So this is when you have a small point change, you get very, very different activity properties. So we created all these data sets internally. And
I just wanted to ask, isn't TDC mainly MoleculeNet, a few extra data sets?
No, we went through all of them. There's some of them contain MolNet, but there are some other ones in TDC.
Okay.
Yeah. So we have the data sets, and then what we wanted to start doing is testing different methods. So we had a way of featurizing molecules from going from a molecule to some representation. So like I said, ECFP, this is more expert features. And then we've got Multivec, which is an older NLP method. And then we go into more of the transformer models like Chemberta. And then some of the architectures that Pietro was talking about, like Informax 2D, when you do large pre-training steps to try and get really strong learned representation. Tried this in the 3D case, and then also other like graph flavors. And then once you've got your representation, you can then start doing some modeling. So support vector machines, there was a paper that came out recently that said, support vector machines rule, stop doing machine learning. So we included this into the benchmark. Tree models that have been shown to be done good on some benchmarks before. Graph neural networks, and then just the standard message passing stuff. And then another one that we included were Gaussian processes, which aren't reported as much as a classical method, but we found them quite interesting, so we also included them. So what did we find? So starting with the featurizers before, like what worked best for different problems. So we grouped into QSAR, which is quantitative structural activity relationships, and then ADMET, which is more of your bio properties. And essentially, what we found is that ECFP fingerprints did extremely well on these activity tasks. So if you have activity data sets, you should probably use expert feature curation for these types of data sets. But when we started looking at biological ones, it was mainly Mordred, again, expert feature, but then we also started seeing graphs pop up as good featurizers, as well as the language models. So I think just in summary, if you have activity data, use classical. If you have ADMET data, you probably need to test a more variety of featurizers. And then what did we see with the models? So again, with QSAR data sets, we found that Gaussian processes by far outperformed any kind of state-of-the-art or other classical methods, which we found really interesting.
Gaussian processes with which kernel?
We mixture. It was in a hyperparameter optimization. I think there was a paper, Gauchi is the library, which built some custom kernels. But in that paper, they referenced only MolNet, so it didn't give you a kind of a sense of how performant they actually were. Yeah. So there's various ones, like you can use Tanimoto, et cetera. Yeah. So I think this is like the first time that we really saw this kind of level of performance from a Gaussian process. And then the ADMET results were really interesting as well. So this is the biological properties. And here we found generally tree models work well, but then Gaussian processes, and then graphs started showing up as well. So it seems that if you're doing activity, you should probably use GPs, but for more biological focused ones, it really depends on the model. So I've spoken so far about like predictive performance. So how good is a model at predicting a single value? And in drug development, there's big bets being placed, essentially. These are like billion-dollar compounds, potentially. It's 2 years of development through different stages just to get the molecule right before it goes into in vitro or in vivo. So if you're making predictions and making decisions and moving forwards, you want to be really careful about which ones you're selecting. Like, is a point prediction good enough? You also want to be able to quantify the amount of uncertainty in a model. So I have two molecules up here, and they have very similar activities, but they have a different standard deviation. So does anybody in the audience want to make a pick? Say you're trying to maximize activity. Does anybody want to take a pick on a molecule that they would go for?
The one on the left.
Why?
What's good?
Wait, one at a time.
I think it might be the chlorine as well, because molecule activity.
Okay, but why would you pick that based, like, why that molecule instead of the other one? Okay, so because there's more confident, in short. So this brings up like the second interesting point, that this depends whether you want to explore or exploit your space, right? So that was a very conservative guess because the standard deviation is smaller. Okay, I want to be really confident. If you're at earlier stages, you might be like, okay, I want to go where there's loads of uncertainty because I might maximize my activity and kind of cover the space more. So we also wanted to get a gauge of like how all the models that we built, like we know how good they are at point prediction, but what does this look like from an uncertainty perspective?
Which one was the best in your opinion, the left-hand one?
I think it depends what you want to do.
I mean, I'm not using AI, I'm using intelligence, because I think the left-hand one is structurally more active.
Uh-huh.
It's based on medicinal chemistry, but I'd be interested to know which one was best.
Yeah, so like the reason why I'm bringing the uncertainty up is that like if you want to make a recommendation, then take a step forwards. Like there's loads of really good algorithms for doing this, like Bayesian optimization, where like computers have like a lot better context of uncertainty. So it really depends on your objective. I would personally say, but then you can also screen, and then you could say, okay, I want to be more Yeah, exactly. And that's like interesting from a patent perspective, right? Is that you may want to search different spaces that you've not seen before and de-risk molecules that you may want to include in a patent application, for example. So like with a similar theme, we went on of like, oh, how do we actually quantify uncertainty in models along with point prediction? And then there's a really cool score called NLPD. I can go into it more afterwards if people are interested. But essentially it measures how well a model is dealing with uncertainty. And essentially what we found, which isn't actually too surprising given that Gaussian processes naturally come with uncertainty baked into them, is that both on QSAR, so activity tasks, and ADMET tasks, Gaussian processes did really well in terms of uncertainty quantification. There is a bit of sacrifice, so sometimes more of the deep learning models do better on these tasks, but then they're not as confident. So you can kind of sacrifice predictive power for having a better understanding of uncertainty. So I'm kind of like coming to the end of the main findings and kind of just wanted to discuss some of the implications and like what I think is coming next. So this image, I asked ChatGPT what the future of drug discovery for AI was, and then I put this. I asked it to generate a prompt. Then I gave this prompt to Midjourney that made this image. And you might be like wondering why I did this. Well, language model, really powerful. Like we have really, really strong learned representations that could describe this image. And then we have really good image models as well, and really good learned representations that enable us to generate an image. And I don't think that we're there in the same way with molecular representation. So I think that there's more work to be done about like how we learn on very, very large data sets in terms of molecules So I think things like exploring, like integrating physics into models and using like equivariant neural networks, et cetera, might be interesting. But I think another huge issue is data. Like ChatGPT had access to the internet. A lot of these models, like Informax 2D, which is graph pre-training, doesn't have access to data at this same scale. And I think this was mentioned in an earlier talk as well, and it's like improving the benchmarks that can push the kind of deep learning technology around molecules or biologics in the same direction that we've seen for image and language models. So yeah, I think we need to work more on the data and integrate more rules of physics into the models. And then just to kind of summarize the general findings of what we found as a company is that not one model works best for any data set at the moment. And I think this is why I think there's maybe more work to be done on the representations. So not one model fits. You should probably test a whole pipeline of models against data sets. And uncertainty quantification is super important. So not just being able to predict, but like knowing how good you are at predicting and having some gauge of that for decision making, because this process, or like how AI is used, or like practically in companies, I think you end up with two cases. You end up with deep research, which doesn't get translated to the end user, like the biologists or the chemists actually making stuff. So you can get this separation, or where it's actually utilized in companies. People are providing lists, so there's a really good synergistic relationship. So like as a company in general, we're trying to bridge this interface and make this accessible so we can actually accelerate the process of discovery. I would just like to say thank you to everybody who was involved with this, especially Jacob, who ran a lot of the machine learning benchmarks. And then Mark, who's at Imperial, he does a lot of Bayesian work. He was really, really helpful and kind of critical in helping us like come up with metrics to quantify uncertainty. And for anybody who would like to test out what the kind of pipeline that we've built that inevitably gets used on users' data, we're currently in closed beta at the moment, so like please reach out to me. We'll happily like onboard you, and you can kind of give the platform a test and compare it to like what your state-of-the-art models are, the state-of-the-art models that you're using at the moment. Yeah, and I'd like to thank the other speakers for the talk so far. It's super interesting and like loads of contributions to the field. Yeah, please reach out if you're interested in the closed beta program, so then you can actually test this and see how it compares to your stuff. And I know this is mainly academic focused, but if anybody's kind of curious about the step outside academia into entrepreneurship, or you're thinking of doing this with your own research, I'm kind of more than happy to speak to you and share some of my own experiences and the journey that we've had as a company. So thank you for listening, and would love to have some questions.